We need to talk about enzymes because we've talked about how we can make endergonic reactions occur and seem like they're exergonic with our analogy of the money and the iPhones and the rotary phones. But another way that cells can be more efficient is by using enzymes, which are these protein machines that make reactions occur more quickly. And they do that by changing the activation energy. So let's talk about what that is. So an enzyme can speed up a reaction by lowering activation energy or lowering the energy barrier. So enzymes are biological catalysts. What is a catalyst? A catalyst is a chemical agent that can speed up a reaction, but it does not get consumed in the process. So basically, the enzyme can perform a function, but it doesn't get used up in the process. It's not like it does the reaction one time and that's it, it's done. Can do it over and over again without being destroyed. So an enzyme is a catalytic protein. So if we put sucrose in water, sucrose is a disaccharide of glucose and fructose held together by that glycosidic linkage. And the enzyme sucrase can break down sucrose very, very quickly. If we put just sucrose in water, it'll break down very, very slowly into glucose and fructose. It'll take months for it to break down without an enzyme there. We add an enzyme and boom, all of that sucrose is converted into glucose and fructose in a matter of seconds. Okay, so you can see here that this enzyme is called sucrase, which is not very different from sucrose. Sucrose is the sugar. Sucrase is the enzyme. Many enzymes end in ASE. That is a clue that you're looking at an enzyme. And then the first part of the name of the enzyme is often whatever the substrate is, whatever the thing that is being affected by the enzyme. So sucrose broken down by sucrase, and a sucrose enzyme. So here's our dimer, our disaccharide with glucose and fructose. In the presence of water, we add the enzyme and it hydrolyzes to form glucose and fructose. So this is a spontaneous process. We could just wait for the sucrose to break down in water, but we don't have to. There's this enzyme out there that can do the job very, very quickly and very, very efficiently. All right, so we're back to talking about energy again. Remember, chemical reactions are the making and breaking of chemical bonds. But in order to do that, we need to stress the bonds. They don't just fall apart on their own unless somehow the bonds are somehow stressed, bent, twisted, folded. Something is done to get that process started. And that energy that does that bending, folding, twisting is called the activation energy. So like when we cook our food, we are changing the things that go into our food by heating them. And that is what we're basically doing. We're causing chemical processes to occur when we cook our food. And we're supplying activation energy. But in living cells, we don't want to cook our cells. We don't want to cook things, but we can do that by using enzymes. So we start off with some initial reactants, say A connected to B, C connected to D. And we want to produce some products, A connected to C, B connected to D, which means we need to break these bonds, the AB bonds, break these CD bonds, and make new bonds, the ACBD bonds. Now, we want delta G to be negative in order for this to be a spontaneous reaction. But in order for the reaction to occur, we've got to stress those AB bonds, we've got to stress those CD bonds. There has to be a transition state. So it requires a little bit of energy to make this transition from these reactants to these products. We call that bump that they have to get over the activation energy, represented here as E sub A, energy of activation. So enzymes lower that barrier, that activation energy barrier. They don't change the Gibbs free energy change. They hasten reactions, they speed up reactions that would occur anyway, but they just make it a lot easier for that to happen. So we add an enzyme, and it turns out that this activation energy can be decreased. It sort of gives the reaction a little bit of a push, which makes it occur a lot more quickly. But the Gibbs free energy of the reactants and the products stays the same because it is a physical property of those molecules. So the initial reactants that are acted upon by an enzyme are called the substrate. The enzyme can bind to that substrate, forming what's called an enzyme-substrate complex. Enzymes are typically very large molecules, and it's not that the substrate can just bind to any old place on the enzyme. It has to find what's called the active site. The active site is where the enzyme actually physically binds to the substrate. And when the substrate binds to the enzyme, the whole enzyme can change shape, which kind of affects that change. So imagine that like you've got a pliers wrapped around a nut. So the nut is kind of like the substrate, the wrench is kind of like the pliers is like the enzyme. In order for that to work, the pliers have to change shape. You have to squeeze the jaws of the pliers closed around the nut in order to twist it. So that's what we mean by induced fit. The shape of the thing changes in order to make that enzyme function. And not everything is going to fit inside that enzyme's active site. So the jaws in this analogy of the pliers are the active site. So here you can see a cartoon of a small substrate molecule like sucrose finding the active site of the enzyme. So the substrate, it's not going to bind up here, back here, over here. It's got to go into this pocket here where it's going to fit. And then the enzyme, like the pliers, is going to clamp down onto that substrate, and the change is going to happen in that active site. So what does an enzyme do that makes that activation energy lower? Maybe it's going to make the two substrate molecules, maybe it's going to combine two molecules together. Maybe it's going to orient them correctly. So, say we want to make sucrose. Well, we have to put the proper parts of the glucose and fructose next to each other. And then maybe they'll just bump into each other. Like maybe if two people just want to hold hands, they have to be next to each other, and the one person's left hand has to be right next to the other person's right hand in order for them to hold hands with each other. If the person is, two stairs up from somebody else, their hand's not going to reach the other hand. Their hand's going to touch their head or something. So we can orient the substrates correctly. We can strain the bonds, kind of like twisting on a nut. Maybe we're going to provide a favorable microenvironment by changing the pH or making it more charged. or they can form a covalent bond to the substrate, kind of holding it in place so that another molecule can come in and join onto it. So here's another cartoon showing how substrates can enter into an active site of an enzyme. Here's the enzyme before it forms the enzyme substrate complex. The substrates are kind of held in that active site, which holds everything in the correct position so that things can change. They can hold hands or switch hands. So the activation energy is lowered, speeds up the reaction. Substrates are converted to some sort of product. So instead of having A, B, and CD, we have AC and BD. The products are released, and now the enzyme is ready to go to work again, but it is unchanged. That's the nature of catalysis, or using a catalyst, is that this enzyme can be reused again and again and again. You can use an enzyme is kind of like a tool. You can use that wrench thousands of times, and you don't need a brand new wrench every time you want to go and tighten a nut, or you don't need to buy a new hammer every time you hammer a nail into a piece of wood. It keeps doing its function, and it's not substantially changed each time it performs that reaction. Okay, so enzymes can be affected by temperature and pH and other chemical factors. So I think this is our last learning objective. How are enzymes affected by temperature and pH? Well, enzymes, being proteins, are made up of amino acids, and they are fundamentally chemicals. And all chemicals are affected by chemical factors like temperature, pH, ion conditions, solvents, things like that. So, an enzyme's activity can certainly be affected by chemical factors like temperature and pH. Every enzyme has optimal conditions for activity. So we can make enzymes work or not work by changing the conditions in a cell or an experimental setup. So what's happening in this chart is, these charts are a couple different things. We're looking at the rate of the reaction. How much is this reaction occurring? So human body temperature, you probably have learned, is 98.6 degrees Fahrenheit, which is 37 degrees Celsius. So almost all of our enzymes are optimized to function at this temperature. There are other organisms, like heat-tolerant bacteria, that can live in hot springs, like in Yellowstone National Park, that are optimized to live near the boiling point of water. Remember, the boiling point of water is 100 degrees Celsius, 212 degrees Fahrenheit. So these heat-tolerant bacteria, they have a temperature optimum of 77 degrees. So you can see, if you have these bacteria, if you were to ingest them, they probably wouldn't be able to live inside your body because their enzymes aren't going to work. At 37 degrees, you can see the activity of this enzyme is close to zero. It's just not going to work. Similarly, your enzymes, if they were somehow put into this bacterium, they're not going to work because they're going to get denatured. The proteins are going to come apart. So every enzyme has a temperature optimum, a best range of conditions for its activity. So right up here is the optimum, the peak, but there's also maxima and minima. Same thing is true for pH, or the concentration of protons in a solution. our stomachs are very, very low pH, very strongly acidic. Now, there are enzymes in our stomach, like, for example, pepsin is an enzyme that is in our stomach that helps break down peptide bonds, breaks up proteins. And it is optimized to work in the very low pH of our stomach. But once things leave the stomach, the pH goes up. It becomes much more basic. So when you go from the stomach into the small intestine, the next section of your digestive tract, our bodies need to raise the pH, reduce the acidity very quickly so that the rest of our body doesn't get eaten by our stomach acid. So in the intestine, trypsin is another digestive enzyme. It works on other types of bonds. So in our small intestine, trypsin is optimized to work at a much higher pH, much less acidic condition, the basic condition, pH 8. So if we were to swap the location of these enzymes, they just wouldn't work. They plain just wouldn't work because they have, based on the types of amino acids that are present in these different enzymes, different optimal pH for them to work. Just like if you hard boil an egg, you are affecting the proteins in that egg and making them from a liquid at normal room temperature. Once you denature them, once you change the shape of those proteins, they become a solid like the egg white and the egg yolk, and they don't go back again. They've lost their function as being chicken building proteins, and they can be more easily digested by you and I. So that's the end of chapter 6. Be on the lookout for, I think, chapter 7 on cell respiration is our next one.